Hello everyone, this is Elongation guy in a new tutorial. In today's tutorial, we're going to cover telomeres. So we're going to understand what is telomere structure and going to cover their function as well. So we will start with the first question. What are telomeres? If we have a look here at the structure, so these are sister chromatids, which are two copies of the same chromosome. And we can see that sister chromatids are connected with a centromere in the middle. These pieces or areas of each sister chromatids are what is known telomeres. So basically, telomeres, they are found at the end of chromosomes in the eukaryotic cell, and in terms of their DNA structure, they are basically containing a repetitive sequence of DNA, usually contains these nucleotides. In terms of their lengths, they range from few thousands to several tens of thousands of base pairs. And in terms of their function, they can act as a protective cap, and also they can induce cellular senescence or apoptosis. And we will go into more depth about these topics as we go through this tutorial. So now let's have a closer look at the telomeric DNA sequence. Okay. So as we know that every sister chromatid or every chromosome is nothing but a single DNA molecule that is highly compacted to form this chromosome. And to get more information on chromosome formation and DNA folding, please just have a look at this video that will appear here, and it's going to cover a lot of the questions you have. So looking at the chromosome in this form, as a DNA sequence, will make it easier for us to understand what the telomeres are and how do they look like. So if we have a look here, we can see that every telomere at the end of every chromosome, it has two portions. It has a portion that is made of double-stranded DNA, and it has another portion which is made of the single-stranded DNA. So the double-stranded DNA region of the telomere is actually making the majority of the telomeric DNA sequence. However, when we come to the single-stranded DNA portion, this portion is found at the very end of the telomere. It also consists of a 3' overhang, so we call it a 3' overhang, and it's only made up of a few hundreds of nucleotides in length. So this overhang is created during DNA replication of the lagging strand, and then the creation of this 3' overhang is a very known characteristic of telomeric DNA, and it plays a very important role in the telomere function and maintenance. And through this tutorial, we will understand how this overhang is very important. So let's first now to have a look at how this 3' overhang in the telomeric DNA is created during DNA replication. So usually DNA replication of the middle portion of the chromosomes start from a replication bubble, as we can see here, and then the replication will move in this way and in that way. And in this lagging strand, these are called the Okazaki fragments. So usually in Okazaki fragments, we would have a portion which is called made up of RNA primer, and then we would have the portion which is a DNA sequence. And what happens is that DNA polymerase will bind to the RNA primer and start sequencing and synthesizing the newly synthesized DNA strand until it reaches the next RNA primer. And at the end of this process, we will have a continuous DNA molecule. So here I'm explaining this process very quickly. To have a more detailed information about DNA replication, please have a look at the video that will appear here. So just keep in mind that this is the DNA replication of the middle portion of the chromosome. So now we will have a look at the DNA replication at either ends of the chromosome. So as we just explained that within the DNA, when the DNA replication starts, it starts from a replication fork, and then the replication fork will be moving outward in these directions. And within that, we will have the Okazaki fragments or the lagging strands, and we would have the leading strand. So our focus here is going to be about the lagging strands, and here's why. So as the DNA sequence as the DNA replication reaches the end of the chromosomes, just pay attention that this is the template DNA and this is the newly synthesized DNA. So this is one end and this is the other end. So at this end of the synthesized DNA, we would have the primer, which was made to make this Okazaki fragment. However, there is no upstream DNA sequence of this primer. And since DNA polymerase only synthesize DNA in the 5' to 3', in the 5' to 3', therefore this RNA will not be able to replaced with a DNA sequence. To get more information on DNA synthesis and how the DNA polymerase moves only on the 5' to the 3', just have a look at this video that will appear here. So what happens is that after these areas, which are the primers, are removed, what we will have is an overhang that will not be replaced with a DNA sequence, because as we said, the DNA polymerase does not have an upstream DNA sequence from which it can extend. And these overhangs are known as end replication problem. And this problem eventually will start what is known as a progressive shortening of the telomere with every cell cycle round. So now let's have a look at the consequences of this telomere shortening that occurs after DNA replication. So the first thing is that usually after several DNA division or cell division, what happens is that telomere will become shortened like that. And usually when the telomere has a normal length, specific proteins such as the sheltering complex can bind to the telomere and protect it from being recognized as a double-stranded break. However, when the telomere is too short, these protein complexes cannot bind to telomere, and therefore it's going to be recognized as a DNA damage. And therefore, what the cell will try to do is to repair this DNA damage. So the exposed ends of the telomere will be recognized as a double-stranded break, and since it's a double-stranded break, the cell will try to repair it using either of two methods, either homologous recombination or non-homologous enjoining. And since sometimes the telomeres will be too short, therefore non-homologous enjoining will be the predominant pathway to repair the DNA damage. However, the problem of this repair mechanism is that it can inappropriately join the ends of two different telomeres, like what's happening here, and this results in telomere fusion. So what are the consequences of such a problem? So first of all, forming the decentric chromosomes, and during mitosis, decentric chromosomes can result in chromosome breakage, can result in further genomic instability or anaphase bridges. Also, it can lead to genomic instabilities such as chromosomal rearrangement, amplification, and deletions. Also, it can lead to cancer. And also the formation of such telomeric fusion might result in cell apoptosis as a protective mechanism to prevent the progression and the division of such a damaged cell. So now we will have a short look at the telomere shortening in cancer cells. So as we now know that in healthy cells, what happens is that after several cell divisions, the telomere will start getting short, and this telomere shortening will induce either cell senescence or just cell apoptosis, and the healthy cell will just die after several cell divisions. However, this does not happen in cancer cells because, as we know, cancer is characterized with an upregulated levels of cell division. So if telomere shortening occurs in cancer, then the cancer cells will not be able to reach and form the tumor. So what happens is that the cancer manages to maintain the length of the telomere without getting shortened after several cell divisions. So now the question is, how cancer can do that? Cancer cells can do that because of their ability of telomere elongation. So how can they elongate their telomere? And this is all come down to an enzyme called telomerase. So as we can see here, we have the 3 overhang here, and this is the telomeric DNA. So what happens is that telomere has an RNA component, which is this component, okay, that contains a short sequence that serves as a template for the addition of the new telomeric DNA. And then it has this other component, which is the telomerase reverse transcriptase, which allows the RNA template to be synthesized into a DNA. So this complex of telomerase is what allows cancer cells to maintain the length of the telomere. So what is the significance of telomere elongation? So first of all, cellular immortality, because cells with active telomerase can divide indefinitely without undergoing senescence. And then also in cancer, approximately 90% of a human cancer exhibit elevated levels of telomerase, making it a potential therapeutic target. And also in aging studies, telomere plays a very important role because it is related to triggering cellular senescence, apoptosis, and therefore contributing to tissue degeneration. And here are some questions to test your understanding of today's tutorial. So hopefully you find them helpful. And with that, we reach to the end of this tutorial. I really hope that you find it beneficial and easy to follow. Please don't forget to like, subscribe, comment, and hit on the notification button so you're going to be notified once we add the new video. Thanks for listening.